This video is for chapter 14, lesson 2. So we're going to be looking at genetic disorders. So let's get started here. So we're going to, in the last lesson, we looked at um, chromosomes in general. We talked about karyotypes, how we can take a picture of our chromosomes. We talked about genetic traits and the different ways that human traits are transmitted. So, like some of our traits are just dominant or recessive, some of them are on the autosome, some of them are linked to the X or the Y chromosome. Today, we're going to look at genetic disorders. Some of them will be linked to just a particular gene, or it could be a chromosomal disorder. We're going to look at a lot of different things today. So we're going to start with looking at how it is that a change in the DNA actually affects your phenotype, right? So remember, phenotype is that physical expression. What do you actually look like? So the DNA, what does that actually cause you to look like? And so by changing the DNA sequence, and we've talked about this kind of periodically all along, that if we change the DNA sequence, that's going to change the protein because it can change the amino acid sequence. Okay, so that's what we looked at more in like chapter 13. And if we change the amino acid sequence, that changes the phenotype. Okay, so let's look at an example. So we've talked about, this was one of the things we did talk about in chapter 13 was sickle cell disease. And we were looking at how it's a change to a single amino acid within that protein. So if you remember, your red blood cells they should look like this. They're kind of disc shaped, but a little concave in the middle. Okay, and this structure is caused by hemoglobin. Hemoglobin is your protein that carries your oxygen. You actually have 4 of them that bond together to make this red blood cell. If you have sickle cell, though, then you have a defective polypeptide. And instead of being this nice round shape, it's shaped more kind of crescenty or sickle shaped. So this hemoglobin doesn't carry the right amount of oxygen, and the molecules tend to clump together. They tend to get stuck, especially in your capillaries. So remember, your capillaries are the really small blood vessels that you have. They're the smallest ones. That's where you actually get the exchange of oxygen and carbon dioxide with your cells. Okay. So by changing a single amino acid in this gene, you end up with blood cells that don't work properly. Okay. Another individual gene. So sickle cell is most commonly found amongst people of African descent. But another disorder that's caused by a single gene is cystic fibrosis, or CF, most commonly found in people of European descent. So most of the time, this is caused by deleting 3 bases, and it ends up deleting this one amino acid right here. So by deleting this one amino acid, this protein that it makes, the normal protein, codes for a membrane protein. Okay, so it's embedded in your cell membrane, specifically in the lungs. And its job is to let chloride ions pass through. If you can't pass these chloride ions, then the result is that you get mucus buildup. And if you know anything about mucus buildup, it tends to lead to bacterial infections. And this is why little kids are real prone to ear infections, because they build up that mucus anytime they're sick. And if you're not clearing that out, the bacteria just like settles in there and it's just like, this is where I live now. And so then you can get ear infections and it can cause all sorts of problems. So people with CF are very susceptible to bacterial infections in their lungs because they have this mucus that they can't clear out. Okay, because these chloride ions aren't able to pass across the membranes of their cells. All because they have one amino acid that's missing from this protein. Okay, so CF, the normal treatment, a lot of times, so they can do like breathing treatments and stuff. They end up hospitalized a lot, um, and eventually they end up needing a lung transplant. So, and um, sometimes a single lung transplant is enough, and other times it's not. And so there are people that have to have multiple lung transplants, and often they don't survive for very long. So I actually knew somebody who had this, and she had her first double lung transplant when she was 11. And she was when she was 17, she was waiting for her second, and they couldn't find a donor in time, so she passed away her senior year of high school from cystic fibrosis. Um, so it tends to be fatal, and fatal fairly young, because you can't breathe. And so even with our medical technology that we have and the ability to do transplants, it doesn't always fix the problem permanently. It's sometimes just a temporary fix. Uh, but anyways, so by deleting this one amino acid from the protein chain, the protein can't function correctly, um, and you end up with CF and bad things. Another example of an individual gene that causes a disorder is Huntington's disease. Huntington's disease is a dominant protein, or so it's a dominant allele. Um, it's not particularly common. It's another example of it's the dominant form, but it's not the more common trait. Unfortunately, you don't usually know if you have Huntington's disease until you are in your middle age. So you've probably already had kids and passed on the gene. Um, so they do have genetic testing. If someone in your family has Huntington's, you can get tested for it at an earlier age. You don't have to wait to see if you have it. But what it's caused by is this repetition. You repeat this CAG in the DNA more than 40 times. Like, it's just this repetition of the same codon. So you end up repeating the same amino acid in your protein over and over again. And by repeating it so many times within that gene, you end up with mental deterioration. So you start to lose your ability to do all sorts of things, just think and reason and be a normal human. Um, and you also end up with uncontrollable movements. It's rather painful. And then you add in like dementia, basically. Um, and so it's not a pleasant disorder to have. And so if especially someone in your immediate family has it, getting tested early is an, not an unreasonable thing to do. But it's just caused by repeating and adding in all of um, the same amino acid, just a big chunk of them all in a row. Um, it does repeat more than 40 times. It's different in different individuals, the actual number of repeats. The more repeats you have, the earlier it tends to um, set in, and the worse the symptoms tend to be. There are some advantages, not so much to having um, Huntington's. Sorry, I forgot which one we just did. Not so much to having Huntington's, but there are some genetic advantages actually to having either sickle cell or CF. So with sickle cell, you are resistant to malaria. So for whatever reason, having those sickle-shaped cells, you don't get malaria. If you are homozygous, right, you have two copies of sickle cell, so all of your blood cells are sickle-shaped, you're kind of, like, oh, well, you may not die of malaria, but you have sickle cell, and it's not really a whole lot better. The advantage comes from the heterozygous individuals. And in genetics, we actually call this the heterozygote advantage. Okay. So heterozygous individuals, remember, would have one normal blood cell copy and one copy of the sickle cell. So about half of your blood cells would be normal, about half would be sickle shaped. So these individuals are generally healthy. They don't have a whole lot of the symptoms of sickle cell, um, because you have enough blood cells that are the correct shape. You're carrying enough oxygen. You're able to, you don't have all of the clumping. You might have some every once in a while. You might be at a little bit more of a risk of blood clots and things like that, but not as much as someone with full on sickle cell. Um, in addition to being generally healthy, you're malaria resistant. Okay. So by having one copy of that sickle cell gene, you're resistant to malaria. So people who live in areas where malaria is a big problem, by being heterozygous for sickle cell, you get an advantage over people who are totally healthy, who have two copies of the normal allele. CF is similar in that you don't get typhoid. For whatever reason, having CF will prevent the typhoid bacteria. It can't get into your lungs and into your body. And so then you don't have typhoid, which again, not a big concern for us in the US, but in a lot of places, typhoid is still a problem. Um, so if you are heterozygous, again, you have enough of the normal proteins that you're able to move those chloride ions across the membrane. So you're healthy, at least largely overall healthy, and you don't get typhoid. So that's where the advantage comes in. And that's where, like, why we still have these things around, these diseases, because being heterozygous is actually more beneficial. So if you have two individuals who are heterozygous, and they have a kid, they could have a kid. If you remember those Punnett squares that we did, they would have a 25% chance of having a child with either sickle cell or CF. Okay, but they would be, the parents would be healthy and have resistance, and they'd have a 50% chance of having kids who are heterozygous and resistant to whichever disease. So there are some advantages to this. All right, the last thing we need to discuss today are chromosomal disorders. So chromosomal disorders are caused by a process called non-disjunction. Non-disjunction happens during meiosis. It can happen during either meiosis 1 or meiosis 2. So this picture over here is showing meiosis 1, where the homologous chromosomes fail to separate. So during meiosis, right, we line up our homologous pairs. Okay, so here they're showing a set of red chromosomes and a set of yellow chromosomes. And then we would distribute, and during normal meiosis, you would distribute a pair of the red and a pair of the yellow, right? Half of this would divide over. So you would have red and yellow and red and yellow. And then each of your cells down here, when you separate the chromatids, would have one red and one yellow. But because of non-disjunction, these yellow chromosomes did not separate during meiosis 1, so we wound up with this cell that has twice as much as it needs. And so when that cell goes through meiosis 2, we still have twice as many chromosomes as we need. Okay, so you end up with an abnormal number of chromosomes. There are only a handful of times, there's only a handful of chromosomes that can go through this non-disjunction and still produce what we would call a viable zygote or a viable individual. Viable meaning they would actually survive. Most of the time, if you have a non-disjunction event, if you have a chromosomal disorder, most of the time that would result in a miscarriage. The few exceptions to the rule, we have trisomy 21. So a trisomy is where you get 3 copies. You end up with 3 copies of that chromosome. So if this is, let's say, an egg cell that has the 2 copies of chromosome 21, and then it would get fertilized by a normal sperm cell, So you would end up with 3 copies because that sperm cell would have another copy of that um, chromosome. And so you'd end up with this cell having 3 copies of that chromosome. Okay, so trisomy 21 gives you Down syndrome, which you're probably at least somewhat familiar with. So that's one where they survive. And for that one, depending on the severity of their symptoms, they can actually survive for quite a while. Um, the other 2 autosomes that are sort of viable, but not really, you can have trisomy of 13 or of 18. And we'll talk about those here in just a minute. Um, and they're viable, but they have some pretty severe symptoms and they don't tend to survive very long. I want to say if you have trisomy 18, the chances of you surviving to the age of 3 is really, really low. So viable, yes, but not really. Um, so having too many chromosomes tends to be a big problem for people. There are exceptions with the sex chromosomes. So if you have only, you can have, it's called a monosomy. So instead of tri, it would be mono here, where you only get 1 copy of a chromosome. The only time that that is viable is if it's the X chromosome. So um, this is called Turner syndrome. You get only 1 X chromosome. There's no Y chromosome. So these people are female, but they're typically sterile. They can have some mental disabilities um, and a couple other symptoms that go along with that. I don't remember all of them. You can actually have a trisomy of the X chromosome. You can have 3 copies of the X chromosome and still survive because they form those bar bodies. Again, I think those people are also sterile. Sterile would mean that they cannot produce offspring. Okay. The other one that your book talks about is XXY. So you get 2 copies of the X and a copy of the Y. So these people are female, or sorry, they're male, but they exhibit a lot more female characteristics. Um, and I think they are also sterile. Don't remember that one for sure. And your book gives a little bit better description of some of their symptoms. I can't remember them off the top of my head, and I don't have your book handy. Um, so chromosomal disorders tend to be kind of bad news. That is the end of the PowerPoint and the notes. I'm going to show you the lab that you're going to do, maybe here in a second. Here we go. So this is the lab that you are going to do. You might end up doing a paper version. So I have a paper version and a digital version. And it's up to Ms. Hartman which one you get to do. Okay. The paper version, you cut apart, they're the same thing. So with the paper version, you have pages that have all of these chromosomes on them. And so you would cut apart all of the chromosomes. You're going to find all of the pairs and match them up and then put them in order for a karyotype. And we talked about karyotypes in the beginning of the first lesson. Okay, so with a karyotype, you find all of your matches of all your chromosomes and you start with the longest ones. Okay, the tallest chromosomes are number 1, and then you go from there. The X and the Y are highlighted for you. X chromosomes are pink and Y chromosomes are blue. So those might be the easiest ones to start with. And then you're going to find your pair. So if you do this in the paper version, you're going to cut them all out. The thing I don't like about the paper version is that if you breathe on these little bitty things, I mean, like they're little. If you breathe on them and they fly everywhere and then you end up losing chromosomes, yeah. Not that that has happened to anyone ever all the time. The digital version is a little better because you don't have that problem. The thing you have to watch out for is that you might accidentally resize some of your chromosomes. So if you notice that you've, accidentally like stretched one out, Control Z and like undo, okay? Hit the undo button. This would probably be easiest. If you do the digital version, I would recommend on a computer rather than an iPad. The iPads will work, but whatever. The other thing that I would recommend doing if you're doing this digitally is once you find a pair. Okay, so this is a pair, right? Because they're the same size. I didn't resize them. And then the striping pattern matches, and then the centromere is in the same place. So these are a match, right? If you select both of them, so you can shift and click both and go to arrange and group, then they are together forever, okay? You can ungroup them if you really want. They don't have to be together forever, but now they're going to move as a pair, which makes it a lot easier as you're going through and putting all of them where they belong. Okay, so match all of them up and then start with the longest ones. The tallest ones are here. What you're looking for is that there are 4 sets of these. I think the directions say you only need to complete 1. If you did another one, I might give you some extra credit or something. So there's 4 sets. They are labeled as A, B, C, and D. You're going to put together the karyotype and then try and diagnose it. So one of them is normal. One of them does not have a genetic condition. The other 3 have one of these 3 possible disorders. So they either have Down syndrome, which is where they have 3 copies of number 21, Edward syndrome, 3 copies of number 18, and Patau syndrome, which is 3 copies of 13. Okay, so that's what the plus means. We have an extra copy of that chromosome. Okay, so as you're going through and you find that you have 3 copies of something and you stick it on number 8, that's not an option. You did it wrong. Okay, so go back in and fix that. Okay, so you should either have normal or 3 copies of 13, 18, or 21. If you get something different, you're wrong. Okay, when you get done with your karyotype, what you would do is answer these questions. Okay, they're not really all that difficult. For the last one, you're gonna Google what the disorder is, and then kind of talk about the symptoms. Okay. Um, so if you do an extra one, you can make a copy of this last slide, and answer the analysis questions again to get credit. Okay, so you have to do the whole thing twice, or you didn't really do anything. But that is the karyotype lab. I think I've allotted 2 days for you to do that. And if you need more, you can take more. Okay. So that's what you're doing, matching up the pairs, and then putting them in order, figuring out where they should all go and what disorder that individual might have. Okay. I think that's everything for chapter 14, lesson 2, and for your lab. Let me know if you've got any questions. I recommend starting on your review because y'all know sometimes you have questions on there. And if I'm not there to answer them, it might take me a little while to get back to you via email. Okay, so email me with questions. And I will see you guys eventually.